Welcome, dear students, to the first subtopic of heredity. In this part of my lecture, I will talk about the Mendelian basis of inheritance. Before we go into the different concepts of the Mendelian basis of inheritance, let us first define inheritance. So, inheritance, or oftentimes called heredity, is the process of passing of traits and characteristics from parents to offspring through genes. And since we will be talking about inheritance, or basically the passing of traits and characteristics from parents to offspring, now let us first talk about the different genetic principles that account for the transmission of such traits from parents to offspring in humans and other organisms. The first inheritance mechanism model is the blending hypothesis. This is the idea that genetic material contributed by two parents mixes in a manner analogous to the way blue and yellow paints blend to make green. This hypothesis predicts that over many generations, a freely mating population will give rise to a uniform population of individuals. However, our everyday observations and the results of breeding experiments with animals and plants contradict this prediction. The blending hypothesis also fails to explain other phenomena of inheritance, such as traits reappearing after skipping a generation. An alternative to the blending model is the particulate hypothesis of inheritance, or basically the gene idea. According to this model, parents pass on discrete heritable units, or otherwise known as genes, that retain their separate identities in offspring. This was documented by Gregor Mendel using garden peas. Gregor Mendel is an Austrian monk who documented the particulate mechanism for inheritance. Mendel developed his theory of inheritance several decades before chromosomes were observed under the microscope and the significance of their behavior was understood. Mendel discovered the basic concepts of inheritance by breeding garden peas in carefully designed experiments. So let's now examine Mendel's experimental quantitative approach. As mentioned, Mendel used garden pea plants in his experiments. The reproductive organs of a pea plant are in its flowers, and each pea flower has both pollen-producing organ known as stamens and an egg-bearing organ known as the carpel. In nature, pea plants usually self-fertilize, which means that pollen grains from the stamens land on the carpel of the same flower, and sperm released from the pollen grains fertilize eggs present in the carpel. To achieve cross-pollination or the fertilization between different plants, Mendel removed the immature stamens of a plant before they produced pollen and then dusted pollen from another plant onto the altered flowers. Each resulting zygote then developed into a plant embryo encased in a seed or pea. Mendel could thus always be sure of the parentage of each new seeds. One reason Mendel probably chose to work with peas is that they are available in many varieties. For example, one variety has purple flowers, while another variety has white flowers. A heritable feature that varies among individuals, such as flower color, is called a character. Now, each variant for a character, such as purple or white color for flowers, is called a trait. Other advantages of using peas are their short generation time and the large number of offspring from each mating. Furthermore, Mendel could strictly control mating between plants. In his experiment, Mendel chose to track only those characters that occurred in two distinct alternative forms or traits. So for flower color, the traits were purple or white. For seed shape, round or wrinkled. And for seed color, the traits were yellow or green. He also used varieties that were true breeding, meaning to say plants that produce offspring of the same variety when they self-pollinate. So now let us familiarize ourselves with the terms that Mendel used in his experiments. Hybridization refers to the process of mating two contrasting true breeding varieties. So the pea generation refers to the true breeding parents. The first filial generation, or F1, is the hybrid offspring of the P generation, while the F2, or the second filial generation, is the offspring produced when F1 individuals self-pollinate or cross-pollinate with other F1 hybrids. So now let us look into the different concepts that Mendel deduced from his experimental model. Around 1860, in a monastery garden in Austria, Gregor Mendel used the character of flower color in pea plants to follow traits through 2 generations. He crossed true breeding purple flowered plants and white flowered plants. The resulting F1, or first filial generation hybrids, were allowed to self-pollinate or were cross-pollinated with other F1 hybrids. The F2 generation plants were then observed for flower color. From Mendel's experiment, when he crossed contrasting true breeding white and purple flowered pea plants, all of the F1, or first filial generation hybrids, were purple. When Mendel crossed the first filial generation hybrids, many of the second filial generation plants had purple flowers, but some had white. Mendel then discovered a ratio of about 3:1 purple to white flowers in the F2 generation. Mendel reasoned that only the purple flower factor was affecting flower color in the first filial generation hybrids. So according to Mendel, the purple flower color was the dominant trait, while the white flower color was the recessive trait. So Mendel deduced that the factor for white flowers was not diluted or destroyed because it reappeared in the second filial generation. Aside from the flower color, Mendel also observed the same pattern of inheritance in 6 other pea plant characters, each represented by 2 traits. So for flower position, the traits included eggshell and terminal, yellow and green traits existed for seed color, and then round and wrinkled traits for seed shape. Now for pod shape, inflated and constricted, and then green and yellow traits for pod color, and lastly, tall and dwarf traits for stem length. So what Mendel called a heritable factor is now what we call a gene. Because of this, Mendel developed a hypothesis to explain the 3:1 inheritance pattern he observed in the second filial generation of offspring. 4 related concepts make up this model, which can be related to what we now know about genes and chromosomes. So the first concept states that alternative versions of genes called alleles account for variations in inherited characters. So for example, the gene for flower color in pea plants exists in 2 versions, purple or white. Now, also, each gene resides at a specific locus on a specific chromosome. This photo shows the alleles, or the alternative versions of a gene. So a somatic cell has 2 copies of each chromosome, forming a homologous pair, and thus 2 copies of each gene. So the alleles may be identical or different. This figure depicts a pair of homologous chromosomes in an F1 hybrid pea plant. The paternally inherited chromosome, which is colored blue, which was present in the sperm within a pollen grain, has an allele for purple flowers, and the maternally inherited chromosome, which is red, which was present in an egg with a carpel, has an allele for white flowers. The second concept states that for each character, an organism inherits 2 alleles, one from each parent. The 2 alleles at a particular locus may be identical, as in the true breeding plants of Mendel's P generation. Alternatively, the 2 alleles at a locus differ, as in the first filial generation hybrids. Third, if the 2 alleles at a locus differ, then one, or the dominant allele, determines the organism's appearance, and the other, the recessive allele, has no noticeable effect on the appearance. In the flower color example, The F1 plants had purple flowers because the allele for that trait is dominant. Lastly, the fourth concept, or the law of segregation. This states that the two alleles for a heritable character separate or segregate during gamete formation and end up in different gametes. Thus, an egg or a sperm gets only one of the two alleles that are present in the organism. This segregation of alleles corresponds to the distribution of homologous chromosomes to different gametes in meiosis. So now let me talk about the Punnett square. The Punnett square is a diagram which is used to predict the allele composition of offspring from a cross between individuals of known genetic makeup. So notice that we use a capital letter to symbolize a dominant allele and the lowercase letter for a recessive allele. In our example of the flower color, capital letter P is the purple flower allele and small letter p is the white flower allele. This photo explains the entire Mendel's law of segregation. So Mendel's segregation model accounts for the 3:1 ratio he observed in the second filial generation of his numerous crosses. It illustrates Mendel's model for inheritance of the alleles of a single gene. Each plant has two alleles for the gene controlling flower color, one allele inherited from each of the plant's parents. To construct a Punnett square that predicts the second filial generation of offspring, we list all the possible gametes from one parent. So here, the F1 female along the left side of the square and all the possible gametes from the other parent, here the F1 male along the top. The boxes represent the offspring resulting from all the possible unions of male and female gametes. So, other useful genetic vocabulary of Mendel's model are the following. Homozygous refers to two identical alleles for a character that are present in an organism for the gene controlling that character. Meanwhile, heterozygous refers to two different alleles for a character present in an organism for the gene controlling that character. Next, because of the different effects of dominant and recessive alleles, an organism's traits do not always reveal its genetic composition. Therefore, we distinguish between an organism's phenotype or physical appearance and its genotype or genetic makeup. In the case of flower color in pea plants, the homozygous dominant genotype and heterozygous genotype have the same phenotype or appearance, which is purple, but different genotypes. This is seen in the figure presented. Note that phenotype refers to physiological traits as well as traits that relate directly to appearance. An organism that exhibits a dominant trait, such as purple flowers in pea plants, can be either homozygous for the dominant allele or heterozygous. So, to determine the organism's genotype, geneticists can perform a test cross. This process was devised by Mendel, which includes breeding an organism of unknown genotype with a recessive homozygote. This is called a test cross because it can reveal the genotype of that organism. In a test cross, the individual with the unknown genotype is crossed with a homozygous individual expressing the recessive trait, which in this case is the white flower, and the Punnett squares are used to predict the possible outcomes. Matching the results to either prediction identifies the unknown parental genotype, which can either be the homozygote dominant or the heterozygote. So in this test cross, we transferred pollen from a white-flowered plant to the carpels of a purple-flowered plant. The opposite or reciprocal cross would have led to the same results. So now let's talk about the second law of inheritance proposed by Gregor Mendel, which is the law of independent assortment. Mendel derived the law of segregation by following a single character. So the F1 offspring produced in this cross were monohybrids, or individuals that are heterozygous for one character. A cross between such heterozygotes is called monohybrid cross. Now, for the second law of inheritance by Mendel, he identified this by following two characters at the same time. So the crossing of true breeding parents differing in two characters produces dihybrids in the F1 generation, which are heterozygous for both characters. A dihybrid cross, which is a cross between first filial generation dihybrids, can determine whether two characters are transmitted to offspring as a package or independently. Gregor Mendel followed the characters of seed color and seed shape through the F2 generation. He crossed a true breeding plant with yellow round seeds with a true breeding plant with green wrinkled seeds producing dihybrid F1 plants. Self-pollination of the F1 dihybrids produced the F2 generation. The two hypotheses, dependent and independent assortment, predict different phenotypic ratios. Only the hypothesis of independent assortment predicts the appearance of two of the observed phenotypes, green round seeds and yellow wrinkled seeds. The alleles for seed color and seed shape sort into gametes independently of each other. So, using a dihybrid cross, Mendel developed the law of independent assortment. This law states that each pair of alleles segregates independently of each other pair of alleles during gamete formation. This applies only to genes on different non-homologous chromosomes or those far apart on the same chromosome, because genes located near each other on the same chromosome tend to be inherited together. So, to give you a summary, in the 1860s, Gregor Mendel formulated a theory of inheritance based on experiments with garden peas, proposing that parents pass on to their offspring discrete genes that retain their identity through generations. This theory includes two laws. First is the law of segregation, which states that genes have alternative forms or alleles. In a diploid organism, the two alleles of a gene segregate or separate during meiosis and gamete formation. Each sperm or egg carries only one allele of each pair. Next is the law of independent assortment, which states that the pair of alleles for a given gene segregates into gametes independently of the pair of alleles for any other gene. So this is the end of my discussion on the Mendelian basis of inheritance. Thank you for watching this video.